Today, we will be discussing as well as portraying the 6 enzymatic situations that occur at the microscopic level. However, before we discuss these scenarios of enzymes, we must ask ourselves, what exactly is an enzyme? Enzymes are proteins that, depending on their molecular structure, control certain actions of the cell. Usually, these enzymes act as biological catalysts, which speed up chemical processes within the cell at an exponential rate and bring about a particular biochemical reaction. These enzymatical situations could be most nearly portrayed by scenarios in the New York City subway, with the inhibitors and substances representing the general people, whereas the active site is the train itself. In this scenario, we have what is known as the enzyme-substrate complex. The substrate, like a regular person, enters the active site, which can be comparable to a subway car. The enzyme changes its physical form as the substrate bonds. Likewise, the substrate itself divides into 2 products when used. Think of it almost as though for every passenger getting on the train, 2 others leave. In this scenario, we are presented with what is known as competitive inhibition, which occurs where an inhibitor and a substrate compete with each other in order to reach the active site first. Our main substrate, presented by light blue, is beaten to the active site by the darker blue, competitive inhibitor. The light blue could represent an individual, whereas the darker blue represents the competitive person who cuts off the passenger to get on the train first. Non-competitive inhibition occurs when a separate inhibitor, represented by purple, binds at a spot other than the active site while still blocking off the substrate, light blue, since this inhibitor would have altered the shape of the entire enzyme, which does not allow the substrate to fit in. This could be comparable to a scenario in which a person gets on the train at a different door before us, thus taking up more space and not allowing us to get on the train. In allosteric regulation, a non-substrate molecule binds or modifies a site other than the active site of an enzyme, which results in the enzyme to change its shape. Allosteric regulation could trigger the activation or inactivation of an enzyme. Think of it almost as though an individual got on the train first, but because someone else squeezed in, the individual is knocked off the train. Feedback inhibition is similar to the substrate-enzyme complex. It begins with one substrate going into the active site and, when used, divides into the usual byproducts. However, one of these byproducts turns itself into a substrate, which is used at a different active site. Think, for every person getting on the train, 2 get off, but one of these 2 people notices that this isn't their stop and gets back on the train in a different spot. In the denaturing of an enzyme, factors such as pH level or perhaps extreme heat cause the enzyme to alter and lose its chemical as well as physical properties, thus dismantling the active site in the process. Consequently, the substrate cannot get in. It's almost as though the train went out of service and closed the doors,